Single ejaculate, volume 2 to 5 ml, sperms only 10%. Color grayish white, opalescent, pH alkaline 7.4, range 7.3 to 7.5, specific gravity 1.028. Composition: fluid from seminal vesicles 60%. It gives milky appearance, contains choline 3F fibrinogen, fructose and flavin, acids ascorbic and citric acid, PGF2. Fluorescence test detects choline secreted from seminal vesicles. Fluorescence UV light, bluish white color due to the presence of choline. Fluid from prostate 25%. It gives mucoid consistency. It gives alkalinity of semen, that is pH 7.4. Contains spermine acid phosphatase, PSA P30, MHS5, fibrinolysin, LDH, Ca, zinc, glucose. Barbario's test detects spermine secreted from prostate. Sperms from testis 10%. Sperm count 50 to 150 million per ml. 90% are motile at the time of ejaculation. 50% are motile at 8 hours. Sterility less than 20 million sperms per milliliter. Speed 1 to 4 mm per minute. Sperm length 55 to 65 micrometers. Sperm head 5 into 3 micrometers. Acrosomal enzymes are hyaluronidase and neuraminidase, zonalysins. Fluid from bulbourethral glands, Cowper's gland less than 1%. Fluid from Littress gland, that is small glands in mucosa of anterior urethra. MLA of seminal stains. In case of sexual offences, that is rape, sodomy, bestiality, sexual homicides, the material dried or wet found at the crime scene or found on the body of alleged accused survivor of rape is collected and examined to prove whether it is semen or something else. In civil cases, for example, legitimacy, disputed paternity, artificial insemination. Semen recovered from vagina. Semen in males is slightly alkaline, pH 7.4, but vaginal secretions in females are highly acidic, pH 3.5 to 4. After ejaculation, the alkalinity of semen neutralizes the acidity of vaginal secretions and thus maintains a pH of 6 to 6.5 in the vagina, at which the sperms become motile, it is 1 to 4 mm per minute, and chances of fertilization are enhanced. When semen is kept at room temperature, 100%, that is full motility, passes for about 3 hours, 50% motility for about 8 hours, and 10% for 24 hours. Sperms may be recovered from vagina up to 24 hours in living and 5 days in dead. Sperm head may be found in uterus up to 2 weeks and in anus up to 48 hours. Collection of seminal stain. We can collect the semen present from vagina, vulva and pubis, inner aspect of thighs, perineum, other areas, for example breast, mouth, hands and abdomen. Clothes, for example panties, undergarments, salwar, sari, dupatta, bedsheet, carpet, used condom and floor. If seminal stain is still fluid, make smear immediately, but if not possible to make a smear, dry the stain to prevent putrefaction. Collection from vagina by a throat swab, pipette aided vaginal washing. Dry stains. By using a wet throat swab, moisten it with normal saline for 30 minutes. Examine under microscope. Scrape with a scalpel blade. If dry stain is present on the cloth, cut a portion of the cloth containing the stain and preserve it. Matted hair can be cut in living and plucked in dead. Wet stains. If a wet stain is present in the cloth, cut a portion of the cloth containing the stain and dry and preserve it. Test for examination of semen. Physical test. Physical characteristics. Just after ejaculation, fresh semen is white or greyish in white or yellowish white, thick, jelly-like, sticky, viscous fluid with characteristic odor. On standing, that is within 30 minutes, its viscosity is lost due to prostatic fibrinolysin. Dried seminal stain on clothes are greyish white with irregular outline and starchy hard in feeling. Examination under UV light. Under UV light, dried seminal stains fluoresce with a bluish white color due to presence of choline in semen. Unfortunately, this fluorescence is not specific to semen, as it is also given by albuminous material, for example, nasal or vaginal leucorial discharge and detergent. Chemical test. Florence test. Florence test is a chemical test for semen described by Dr. Florence. Procedure. Step 1. Put a drop of watery solution of seminal stain on the slide. Put a cover slip on it. Allow a drop of Florence reagent to run under the cover slip. Wait for few minutes, observe under microscope. Dark brown rhombic needle-shaped crystals are found. Reaction. The fluorescence reagent Ki plus I2 plus H2 reacts with choline secreted from seminal vesicle and gives out choline periiodide crystals. Choline periiodide crystals resemble Semen crystals. However, choline periiodide crystals are larger and arranged in the form of clusters, rosette or crosses. This test is not specific for semen. Some animal and vegetable substances also give positive result. A negative reaction proves that stain is not seminal. Choline is responsible for the bluish-white fluorescence under UV light. Barberio's test. It is a chemical test for semen described by Barberio in 1905. Procedure. Step 1. Put a drop of watery solution of seminal stain on the slide. Add a drop of alcoholic solution of picric acid to it. Put a cover slip over it. Allow to mix slowly. Wait for a few minutes. Observe under microscope. Yellowish long needle-shaped crystals. Reaction. The alcoholic solution of picric acid reacts with spermine secreted from prostate and spermine picrate leading to spermine picrate crystals. Puranan's test is similar to the Barberio's test, but here dinitro-naphthol sulfonic acid is used instead of picric acid and the crystals are orange colored. Seminal acid phosphatase SAP test, Walker test. SAP is secreted by the epithelial cells of prostate, that it is specific to males. The level of enzyme is equally high in infertile males and vasectomized males. Its secretion begins in males at puberty. Its level remains high until 40 years. Thereafter, its level declines. SAP activity with time. Acid phosphatase activity is 500 to 1000 times greater in human semen than in any other fluid in the body. In a seminal stain, SAP activity decreases with time and activity may be detected for months. Activity is lost due to putrefaction. Levels of SAP in semen: 20 sigma units per milliliter, more than 5 Bodansky units per milliliter, 25 King Armstrong units per milliliter, or 2 international units per milliliter. Procedure: make reagent by mixing alpha naphthyl phosphate and brentamine fast blue B. A cotton swab moistened with sterile water. Touch the suspected stain with cotton swab. Add reagent to swab. It gives out a purple color developing within 2 minutes, which is positive for semen. Any color developing after this is negative. This test is sensitive but not specific for semen because SAP is present in other body tissues also. For example, vaginal secretions, RBCs, animal semen and vegetables like cauliflower, ginger, clover, turnip and raisin. Time since intercourse. SAP levels in vagina falls with passage of time, so its level may indicate time since intercourse. Levels more than 138 sigma units per ml confirm that sexual intercourse occurred within 24 hours. 50 to 138 sigma units per ml is highly suggestive of sexual intercourse within 24 hours. 20-50 sigma units per ml suggests intercourse within 48 hours. Less than 20 sigma units per ml suggests absence of semen. Prostatic specific antigen PSA P30 is a glycoprotein secreted by epithelial cells of prostate. Thus it is specific to males, semen, urine and blood. It is also known as P30 because its molecular weight is 30 kDa. The level of PSA is equally high in infertile males and vasectomized males. Initially it was considered to be specific to prostatic specific, but now it is known to be present in lower concentrations in various body fluids, for example breast milk, amniotic fluid. It can be found in vagina up to maximum of about 2 days. Creatine phosphotokinase. Its concentration is double in sperm than in other body fluids. It is quite stable and the test gives a positive result in stains as old as about 6 months. It is also found in other tissues, for example brain, muscles, retinal photoreceptor cells. FISH, FISH, fluorescent in situ hybridization. After a normal ejaculation into female genital tract, sperms can be identified up to only 2 weeks, but some Y chromosome specific cells from penile epithelium, can be detected in vagina up to 3 weeks after coitus by FISH. So in case accused was vasectomized or did not ejaculate, FISH can be performed on vaginal swabs. This technique detects and localizes the presence of specific DNA sequence on chromosomes. Fluorescent probes that bind to specific sequence are used and detected by fluorescence microscopy. Immunological tests MAB4E6. The epithelial cells of ejaculatory duct secrete an antigen 4E6 which binds to the surface of spermatozoa. This antigen is detected by monoclonal antibody MAP4E6. MHS5 monoclonal mouse anti-human sperm antibody detects SVSA, seminal vesicle specific antigen produced in seminal vesicles, obsolete. Ammonium molybdenate test detects phosphate in seminal fluid. Microscopic test, confirmatory test for semen. Presence of spermatozoa under light microscopy is confirmatory. The sperms are seen motile in wet specimen and non-motile in dry specimen. Wet specimen. Remove semen by a moist swab and then touch it on a glass slide. Put a drop of normal saline on it. Examine under microscope for motility. At room temperature, motility persists for about 3 hours. 50% are motile for 8 hours. 10% for 24 hours. In living persons, motile sperms may be seen up to 6 hours after ejaculation into vagina. Complete sperms may be seen maximum up to 3 days, after which they disintegrate. Next, dry specimen. Example on a fabric. Put a few drops of 1% HCl or 3% acetic acid on the dry specimen and wait for 1 hour if stain is fresh and 4 hours if it is old. Then rub the moistened portion of fabric slide on a glass slide. Dry in air, fix it for 1 minute in methyl or ethyl alcohol. Then staining is done by any one of the following methods. 1. Christmas tree stain. This stain consists of 2 dyes, that is nuclear fast red dye which stains the nucleus red, and picro indigo carmine dye which stains other cells, that is epithelial cells green. Methylene blue for 30 minutes and counterstain with eosin for 2 minutes. Hematoxylin for 2 to 5 minutes and counterstained with eosin for 3 minutes. Only posterior 1/3 of head containing nucleus and the tail is stained by eosin, leading to deep red or pink. The anterior 2/3 of part of head acrosome is faintly stained or even not stained.